Hi, Mr. Crane again. You guys going to get tired of seeing me. So we're going to go over scientific methods in action. I pulled it up on the screen. I'll probably have to do this in two short sections. We'll do bearberry first, and then we'll do staphylococcus second. So real quick, so I'm going to read this for you. So in 1887, a strange nerve disease attacked the people in the Dutch East Indies. The disease was bearberry. Symptoms of the disease included weakness, loss of appetite. Victims often died of heart failure. Scientists thought bacteria might have caused the disease. They injected chickens from bacteria from the blood of patients with bearberry. The injected chickens became sick. However, so did the chickens that were not injected with the bacteria. One of the scientists, Dr. Eichmann, noticed something. Before the experiment, all the chickens had eaten whole grain rice, but during the experiment, the chickens were fed polished rice. Dr. I don't know how to say that for real. Eichmann researched this interesting case. He found that polished rice lacked thiamine and vitamin necessary for good health. So the first thing it asks you is to state the problem. So this is real simple because all you have to do is go up there and you cut and paste that in this particular section right here, and there you go. That's the disease. That's what the problem was. So they had a hypothesis again, another quick um, cut and paste. They thought the scientist that a bacteria caused a disease. And then how was it tested? So again, another one. They injected. Sorry, lunch is over. They injected chickens with bacteria from both patients with bearberry. The injected chickens both became sick. However, so did the other ones. So their hypothesis that they originally thought about the chickens getting sick wasn't correct because both injected and not injected got sick. So should you support or reject that particular um, hypothesis? Well, it wasn't. It didn't pan out the way they thought, so you should reject it. And then what should the new hypothesis be? So what would the new hypothesis be? Well, you would give both chickens the same type of rice, inject one group and not the other group, and then see which one of them survive. So you can try it with the whole grain rice and then try it again a second time with the polished rice. I'm sorry, the polished rice, and probably the ones with the polished rice are the ones that are going to die. So that's the new hypothesis. Now you need to identify your independent variable, dependent variable, constants, and controls. From yesterday's lab or yesterday's session, you learned that the independent variable is something the scientist is changing in the experiment. So what are they changing? Well, or they should have only changed one thing. You found out that they changed two things in here. They should have just changed the chickens that were getting injected versus the chickens that weren't getting injected, or kept the rice the same and give both the injections, and then change the rice the next time and you do whole grain rice, and then everybody gets the injections, but change whole grain rice and those that had polished rice. And that's probably the best way to do it. So independent variable would be the thing the scientist is changing. So that would be the rice. You need to change the rice in the valid test. Dependent variable is the things we're measuring. So we're measuring the death rates of those chickens. Sucks for the chickens. I hate that. But if it's going to save lives, then that's why they test it on animals before they do people. Constants. Three things that you're keeping the same. What are you keeping the same here? Well, I'm keeping the chickens are the same. The injections are the same. The times the chickens are given food could be the same. Maybe the locations are the same. So many different things. And the control is a group that you compare your group that you're testing to. So if you're going to have a group that you give whole grain rice and one that you give white rice, polished rice, and they all get injections, then the ones that you think are going to be just normal, maybe the whole grain rice, that's going the whole grain rice and injected, or the white rice and injected. One of those groups will be the comparison group. So you just have to pick which group, and then which group is going to be your experimental group. So whole grain rice is one your hypothesis is that you think whole grain rice contains thiamine, which is necessary for good health, then that's going to be your experimental group. All right, so hope that was helpful, and we'll stop and do Staphylococcus later.